Welcome to the front row. My name is Jamie Williamson. I'm Executive Vice President here at Scripps Research. And today we're pleased to have Katya Lamia, who's going to be telling us about circadian rhythms. Now, I thought I would maybe say a few things, maybe in general about Scripps Research. Some of you may not be so familiar with this, but we are one of the largest independent, not-for-profit biomedical research institutes in the world. And we have a very interesting culture because we have only researchers without any undergraduate education. And there's a few ways that we try to impact science. And one of them is to really create a silo-free environment where people can do interdisciplinary work. So we don't have explicit teaching requirements, although we do have a world-class graduate program. And we like to say, hey, there's no bad ideas. So we like to have a really open dialogue about science. Another strategy that we use here is that we really try to have a diverse group of scientists. We have graduate students, we have junior faculty, there are young scientists that just emerged from their postdocs. And then we have some really amazing world-class researchers that are thought leaders in their respective fields. And so we're happy to enjoy this environment, and it's turning out to be a real magnet for the top scientists from around the world to come here. And so we really have this amazing, diverse international community that brings all sorts of training and perspectives to the campus. I'm just not going to read all of these, but I just want to throw this up and give you an idea. These are the universities that our current faculty attended to get their training. Jing-Kuan Yu is an incredible chemist, and he was University of Cambridge. Silke Paust is a German scientist by heritage who got her PhD at Harvard. Luc Tétan is an outstanding immunologist who was trained in France. Donna Blackman is the chair of chemistry, and she actually was recruited here from Imperial College in London. Maybe I think we have another set of people, and we just wanted to highlight, we had some big news a couple weeks ago. We were thrilled. Barry Sharpless was awarded, he was one of 3 people to be awarded the Nobel Prize in Chemistry, and the most remarkable thing is that this was his second. And again, we have scientists from all over the world. These are our faculty that we've recruited from various universities. So today is Katya Lamia. And Katya is an outstanding biophysicist by training, and she's now using her skills to understand circadian rhythms. And that's the subject of her lecture today. Now, maybe I'll say just a little bit about this. I think everybody here that's listening fundamentally understands what circadian rhythms are. It's the daylight cycle. Everybody, you get up in the morning and you go to sleep at night, and your day is organized around this rhythm that's been going on ever since humans started walking this earth. And so it may not be surprising that some aspects of that day-night cycle are encoded in our genes in very interesting ways. And we all have this visceral understanding of what happens when something is off with your circadian rhythms. One of the kind of annoyances of United States life is we have this peculiar thing called daylight savings time. And you're just shifting by 1 hour twice a year. I mean, it's really annoying. Your clock is off, it gets dark early, you have to get up too early. Everybody gets that. The other thing that everybody has experienced is jet lag. So now you're traveling multiple time zones and you have to get up early or go to sleep early, and it's fundamentally disruptive to you. So everybody understands this. So what we're going to hear today from Katya is really the history of how people came to understand the genes that do this, and a little bit about how that day-night cycle is regulated, and then some really interesting new results that are going to surprise you about the consequences of disrupting your circadian rhythms for human health. So I'll stop there, and in the time remaining, Katja is going to give us her talk. Katja, welcome.
Thank you. Thank you very much, Jamie. And thank you everyone for being here and giving me the opportunity to share with you today my excitement about circadian biology and what it can teach us about how we interact with the world around us. So first, I'm going to start off just a little, very tiny peek into how I got interested in this area of research and kind of my background. So I grew up with a lot of siblings in a family that has roots in fishing and boat building industries a couple generations back. And in a few slides, you may see how that relates to circadian rhythms. As Jamie mentioned, I studied physics. I followed in my maternal grandfather's path into studying physics as an undergraduate, and then kind of following a side path into biological science in my case. And he ended up studying medicine and practicing for a couple decades before he retired and returned to his hometown of South Bristol, Maine, to farm oysters. And I have the pleasure of visiting my parents there nowadays. I got my PhD in biophysics at Harvard, and then I did postdoctoral work with Ron Evans at the Salk Institute down the street, which brought me here to Scripps finally about 12 years ago. So I want to share with you a couple of take-home messages that I hope you can leave this talk with today if you take nothing else. Circadian clocks are present throughout your body and they promote health. Disruption of circadian rhythms, such as it happens with shift work or even jet lag or time changes, as Jamie mentioned, can actually disrupt your physical health and increase the risk of some disease processes. And in my lab at Scripps, we're trying to understand why this happens and just trying to understand at the molecular level the mechanisms that allow the different cells of your body to keep track of the time of day and how that interacts with other physiological systems to promote health. I'm going to give you just a brief outline of how the talk is going to go to help you follow along. So I'm going to go through a little bit of history of circadian clocks research. Since many of you are probably not familiar with that, I want to give you a little bit of background of how this field developed over time and how I got interested in it. And then I'll tell you some of the early work I did when I first started studying circadian biology. And then finally, I'll tell you about a recent story from some work that was done in my lab at Scripps. And then finally, I'll save some time for questions and answers at the end. So I'll start off with a brief background and history of circadian biology research. So biology in general is really all about how we as organisms and other types of species and organisms interact with the environment around us. And how do we maintain the appropriate internal conditions for health in the face of fluctuating conditions in the external environment? And so, as Jamie mentioned, normally you probably don't think at all about the challenge posed by daily fluctuations in your external environment that come from the rotation of the Earth. But you do notice that when you're out of sync with those rhythms, such as when you experience jet lag or even during twice yearly shift in our clock time, you notice that you feel off. And so that kind of highlights the importance and the normally very smoothly functioning systems that allow you to stay in sync with those external rhythms. And so I'm going to tell you a little bit about some of the research that led to the understanding that there are actual internal timers in all the cells of your body that help maintain that synchrony and some of the things that they do. So circadian biology research really started with a study of plants. And I'm showing here on the right a video of a sunflower with its leaves moving across the course of the day to optimize their exposure to sunlight. So you probably know that plants get their energy from photosynthesis, and that comes from sunlight. And you can see that at night, the plant leaves droop and it almost looks like it's falling asleep. But what's really remarkable is you can see the leaves starting to move toward the east before the sun rises. So it's not simply a response to the light, but that's evidence that the plant is somehow able to keep track of the time and anticipate sunrise in order to and move the leaves towards the direction that it expects the sun to be appearing. Um, and it might seem strange now, but this was a really hot topic in biology in the 19th century. Even Charles Darwin wrote a book about the movement of plant leaves, shown here on the left, although it's less well known than some of his other accomplishments. This was, kind of an enduring mystery that was very of great interest to biologists at the time. So as I mentioned, and you can see in this video, the plant leaves move to maximize their exposure to sun across the day, and they anticipate sunrise, suggesting that there's an internal timer. So the first time that I was actually introduced to the study of circadian rhythms came from a class that I audited my first year in graduate school. So I had studied physics as an undergraduate, so I was trying to really increase my understanding of biology in general as a first year graduate student in the biophysics program. And I had never heard of circadian rhythms before, but this idea that there might be kind of internal and external cycles that needed to be synchronized was very appealing to me with my background in physics at the time. And one of the organisms that was a focus of this class is a phosphorescent algae that you might have seen. It's called the sparkle of the sea. So you may have seen this phosphorescence in the ocean, and it was something that I was really familiar with just personally growing up in my family and spending a lot of time in the water. And it's really beautiful. And you draw your hand through the water and these things sparkle. And what I came to learn in this class is that that's actually coming from an aquatic organism, a marine algae that emits light. And that's actually a defense mechanism for the organism. It allows them to camouflage to their predators that are coming from beneath. And the sparkling makes them camouflage with the surface of the water that has moonlight reflecting off of it. And these algae only emit this light at night. And there was a professor, Woody Hastings, at Harvard, who studied this phenomenon and demonstrated that it was regulated by a clock that was present in these algae that regulated the timing of this light emission. So that kind of sparked my interest in the field. When I give talks to people in California, I like to mention there's also a local example. There's an organism called the California grunion that lays its eggs precisely at the time of the very highest tide. They lay their eggs just above the waterline. And the fact that they do this at the highest tide of the month is really critical because it allows those eggs the time to hatch and the babies to develop enough before the tide gets that high again and washes them back out to sea. And I found this just amazing that these animals are able to somehow figure out when that's going to be and follow this precise timing. And I have to mention that many aquatic organisms in a similar way integrate these sort of daily and monthly cycles to regulate their behavior, not only to the time of day, but also to the specific time of month in sync with the tides. And these, the clocks that allow them to keep track of the tidal times is actually still somewhat mysterious. And it's a topic of intense research, not in my lab, but in other labs. And I'm excited about that just because it's this enduring mystery. But what I'm going to tell you about today is more related to daily rhythms. And so now I've mentioned these two aquatic organisms, but even, pretty much every organism that has been studied in detail has been found to have some sort of circadian daily regulation of its biology. And even humans, even for people, there's a lot of aspects of our physiology that have been shown to follow these kind of rhythmic patterns. And there are times of day when different aspects of your physiology show peaks and troughs, some that you may have some experience with or kind of recognize or be more aware of. Your body temperature actually has a very strong circadian rhythm that's very regular. Your body temperature changes by 2 or 3 degrees every day, and your highest body temperature occurs in the evening, and the lowest occurs early in the morning. And I'm going to come back to that later. Also, several aspects of your exercise physiology change with the time of day. And people who've studied this have shown that, for example, Olympic records tend to be set in the evening because that's when the greatest cardiovascular efficiency and muscle strength occurs. But going back to our historical perspective, I'm going to tell you a little bit more about how mammalian circadian rhythms were proven to be driven by an internal biological timer, and how the components of those timers were discovered. So, after all this work on plants, and there were rhythmic phenomena that were described in many species, including the ones that I've mentioned already, there was a biologist named Colin Pittendrigg who was at Princeton in the 1960s, and he did these really seminal experiments, very carefully and quantitatively documenting behavioral rhythms in rodents. And so here I'm going to show you how that works and what some of the data looks like. Here I'm showing what we, and in my lab, we also do a lot of work with mice. And if you've ever had a pet mouse, hamster, gerbil, you may have given them a running wheel, and you know that they really love to run in these running wheels when they have access to it. And in the lab, we give mice access to a running wheel. We can hook up an electrical circuit to the wheel such that every time the wheel spins, it closes an electrical circuit, and we can record that, that movement. And the data looks like this. So when the mice are resting and not running in the wheel, we don't see anything. It's just blank. When they are running in the running wheel, we see a black tick mark, and the height of the tick mark is related to the number of times that the wheel spins during a given time period. So here's what some of the data looks like. Mice are nocturnal, so they're running when it's dark. And you can see, they're not moving continuously, but it's very consolidated to the dark phase or nighttime period. And here, the way we present this type of data, we show a black bar when the lights are off and a white bar when the lights are on. And you can see that they're very active during the dark phase. What's interesting is when you just turn the lights off, so they no longer are getting a timing cue from the light. If you just keep the mice in constant darkness, they continue to exhibit this rhythmic behavior, but the period is slightly shorter than 24 hours. So the data looks like this. It kind of drifts a little bit earlier every single day. And this data was a really important discovery in this field, because the fact that this rhythmic behavior continues when there's no cue to the time, but it's slightly out of sync with the Earth's rotation, was a really strong indication that this behavioral rhythm is coming from an internal timer, because any signal that might be coming from the external environment to initiate this behavior would have to have a precise 24-hour rhythm. And the fact that Dr. Pittendrigg and his colleagues could measure this continuing rhythm that was a little bit different from 24 hours demonstrated was a very strong indication that this was not coming from any signal from the external environment. And we use this term circadian to describe these types of rhythms that are close to 24 hours, so circa about, and DN about daily. And this term is used to describe these rhythms that you can observe in behavior and other processes that are close to 24 hours. So this type of data can also be collected from other species. And the fact that it's so quantitative and so precise was a motivation for Ron Konopka and Seymour Benzer to use this type of analysis to study behavior in fruit flies and to try to identify genes that could influence behavior. So this was a really big question in neuroscience in the 70s. And what I'm showing here is some of the data that they collected from fruit flies. It's very similar to what people do with mice and running wheels, but in this case, the flies are running through a little glass tube, and they record every time the fly crosses the middle of the tube and breaks an infrared beam. And that's what these little black tick marks are indicating. And so at the top here is recording behavior from a normal fly. You can see they have a rhythm that's with a period that's slightly longer than 24 hours. Ron Konopka brought the idea to Seymour Benzer to use this as a quantitative measure of behavior that could be used to look for genes that influence behavior. And that screen was successful, and they identified several different genetic mutants of fruit flies that had altered behavior, either no rhythm at all or a shorter than normal period or a longer than normal period. And those discoveries, first of all, it was really exciting because it was the first demonstration that a single mutation in a single gene can control behavior. And also, those mutants that they identified led to the discoveries by several groups that actually identified the specific gene that was mutated in those studies. And they were able to restore rhythmic behavior in the mutant that didn't have any rhythms by simply fixing this one single gene. So this really showed that this one single gene could be responsible for driving these behavioral patterns. And then the identification of this gene, which they named period, allowed them to understand the molecular nature of circadian clocks that were driving these behavioral rhythms. And I'll talk about a little bit more about that in the next slide. Circadian rhythms involve, they have to have a mechanism that can generate rhythmicity. And once these, the different components of circadian clocks were discovered through the genetic experiment I just described, similar genetic experiments in other organisms, or by based on the similarity of other genes to those genes or interaction of other proteins with the proteins that were identified through those genetic experiments. All the different components of the circadian clock were discovered. And then through early studies of their functions, it was determined that they form a feedback loop, a negative feedback loop, which makes a lot of sense because, as you can imagine, a lot of different rhythmic behaviors, rhythmic phenomena require this sort of negative feedback. Here I'm showing as an analogy an electrical circuit that involves, a positive factor that drives an activity that represses that original positive factor, so that A turns on B and B turns off A, resulting in this sort of rhythmic activity of factor A. And it's a similar structure for circadian clocks. And you might have heard of the central dogma of biology, in which genes that are present in DNA are converted into RNA through a process called transcription. And that RNA is converted into proteins, which are kind of the active factors, through a process called translation. And there's a certain family of proteins that are called transcription factors. These proteins interact with DNA and catalyze its transcription into RNA. And there's also a class of proteins called transcriptional repressors that interact with the transcription factors and inhibit their activity. And it turns out that the components of circadian clocks are transcription factors and transcriptional repressors. And in mammals, including humans and mice, which are studied in my lab, the transcription factors that are part of the mammalian circadian clocks are called clock and BMAL1. These two proteins interact with each other and turn on the expression of thousands of genes, including their own transcriptional repressors, which include the period protein that was discovered through the Drosophila, the fruit fly genetics that I described, and another group of proteins called cryptochromes or CRYs, which we also study in my lab, but I'm not going to talk a lot about them today. So once these components, the sort of gears of circadian clocks were discovered, it led to new ways of studying the function of circadian clocks. And at that time in the 90s, there was a cutting edge technology that was basically making use of proteins that emit light. And I'm going to briefly tell you about two different proteins that emit light that were then used in elegant experiments to get further understanding of how circadian clocks act throughout the body. So you probably have seen fireflies, their tails light up at night, and that is coming from a specific protein called luciferase that emits light. Another example, there's a protein called green fluorescent protein that comes from jellyfish. So again, back to our aquatic organisms. And this protein emits green light, and both of these can be used in biology to actually watch what's happening in real time. And this was taken advantage of by several groups studying circadian biology. Steve Kay's lab initiated these experiments, and one of the things they did was to make a fruit fly that expressed that green fluorescent protein from jellyfish connected with the PER protein that was discovered in the Seymour Benzer and the Nobel Prize winning studies that I mentioned from Michael Rosbash, Michael Young, and Jeff Hall. And so basically, Steve Kay's group expressed this green fluorescent protein under the control of the elements that normally regulate the expression of PER, so these circadian elements. And their goal was to understand how behavioral rhythms were generated in the brain, but they were really surprised to see that green fluorescent protein controlled by these circadian elements was not only present throughout the body, but it was rhythmically expressed. So they could see these flies lighting up and getting dark again, regularly every day. And that was a really big surprise. And then even more surprising, they could take biopsies of different tissues from the flies and grow them in a petri dish, and they could still see these rhythmic light emissions. And that would continue for weeks. And similar experiments were done with a mouse using the luciferase protein that I described from fireflies. And so this was a really big surprise, showing that circadian clocks were present throughout the body, both in flies and in mice. And so that was kind of the state of the field when I finished my PhD studying insulin and metabolic physiology. And I had just kind of maintained an interest in circadian biology since I first heard about it in my first year in graduate school. And I decided that I really wanted to work on understanding whether circadian biology might actually regulate insulin signaling and metabolism. And so here I'm going to show a little bit of data from one of my papers. And this is a quote from Colin Pittendrigg, who I mentioned was one of the people who really did the seminal work that proved that circadian rhythms are driven by an internal timer. And what he said is a tongue-in-cheek response to Gloria Steinem. He said, a rose is not necessarily and unqualifiedly a rose. That is to say, it is a very different biochemical system at noon and at midnight. Making the point that because there are circadian rhythms throughout every cell of the body, and also in plants, so in every cell of the rose is quite different at noon and at midnight. And here I'm showing an example from one of my papers. These little black images are showing the measuring these proteins that are named on the right-hand side in biopsies from mouse livers that were collected at different times of day. So the numbers at the top represent the time of day. It's actually, the number is the number of hours after the lights were turned on. So the samples under the yellow bar are during the daytime, and under the black bar are at nighttime. And so you can see for cry and purr, these are some of the proteins that are part of the circadian clock. Of course, their expression is very different at noon and at midnight, but also these other proteins that I won't go describe in detail, but reverb alpha and AMPK subunits of AMP kinase, these are really important regulators of metabolism. And you can see that their expression is also different at noon and at midnight and at other times of day. And so this suggests that really all aspects of biology might be under the control of circadian clocks. And so, after the discovery that circadian clocks are everywhere and they regulate the expression of different genes, the big question at this time that I was starting to work in the field was, what are circadian clocks doing in all these different locations? So I showed on the last slide that the amount of different proteins is different at different times of day. And one of the first things that came from many different labs at the time, this was in the early to mid 2000s, many different labs used what was a cutting edge technology at the time called microarrays to look at the expression of all the genes in the genome at different times of day in many different types of tissue biopsies. And all of these studies came to the same conclusion that a very large fraction of the genome undergoes these rhythms in expression and production of their protein products, basically, resulting in very different protein component in different cell types at different times of day. And here on the left, I'm showing an example. This actually comes from a recent paper from my lab, but it's just to illustrate this point that was understood a long time ago. Here I'm showing that these are all of the genes. So each horizontal line is representing a specific gene, and each column is a sample of in this case, it's a muscle biopsy that was taken at a different time of day. Again, the yellow bars are when the lights are on, the black bars are when the lights are off. And this is two different groups of mice that were either sedentary, so they don't have a running wheel in their cage, or active. They do have a running wheel in their cage. And in this case, in this example, what we were showing is that their behavior actually influences how gene expression is modulated by the circadian rhythms. And when the mice are active and running, in this case, the muscle is very responsive to that activity, and they have more rhythmic gene expression in the muscle when they have a running wheel in their cage. And in this case, there are about almost 4,000 genes exhibit these rhythms of expression in mice that have a running wheel, which is about 25% of the genome. So you can see it's a really significant proportion of the genes. Here I'm showing data from another group. This is from a study from John Hoganesh's lab that was published in 2014. And these are basically Venn diagrams showing that, although any particular tissue type that's analyzed has a high percentage of the genes that exhibit these rhythms of expression, but which genes are rhythmically expressed depends on what organ is being studied. And these Venn diagrams are showing the overlap of which genes exhibit rhythmic production in the liver or in these other tissues. So for example, there are a little over 3,000 genes that are expressed rhythmically in the liver, and almost 3,000 genes are expressed rhythmically in the kidney, but only about 600 of those genes are expressed rhythmically in both of those organs. And the only genes, I don't remember the exact number, but it's very small, something like only 10 genes are rhythmically expressed everywhere they looked, and those are the core clock genes, like the period gene. But if all of the tissues that they looked at, if they take all of them together, more than half of the genes in the genome exhibit these rhythmic expression patterns somewhere in the body, including many genes that are important targets of widely prescribed drugs or very important in different disease processes, suggesting again that circadian regulation of physiology might have really important implications for health and disease. Okay, so now I want to get back to kind of the big picture. And here I'm showing a picture of Claude Bernard, who was a French physiologist in the 19th century, who he coined the concept of homeostasis, basically the idea that an important goal of biological systems is to maintain a constant internal environment in the face of challenges from the external environment. And I got very interested in the idea that these, daily predictable fluctuations in the environment present a challenge to homeostasis. And I wanted to ask how circadian clocks can contribute to managing that challenge. And so I'm going to very briefly describe, this was the work I did with Chuck Weitz at Harvard Medical School. So when I finished my PhD, I went to work with Chuck Weitz to ask whether circadian clocks can influence metabolism. And the approach we took to do this, I won't describe in technical detail, but basically we generated mice that do not have a functional circadian clock only in their liver. So they have totally normal behavior. They're eating and active at the normal times, and they only lack circadian rhythms in their liver. And one of the major functions of the liver is to regulate your blood sugar. And so we measured blood sugar, also called glucose, at different times of day. And keep in mind that mice are nocturnal, so they're resting during the day, which is again shown by this yellow bar, and they're eating at night, this black bar. And when we measured their blood glucose, and the normal mice are shown with these black bars and symbols, and the mice that do not have a clock only in their liver are represented by the red bars. And you can see that the mice that don't have a clock in their liver experience this drop in blood sugar during the day when they're resting. So that suggested to us that the presence of a circadian clock in the liver basically enables the mice to predict this time of daily fasting and increase the production of blood sugar, of sugar, and increase its production and also its export into circulation. And we measured the expression of some genes that are known to participate in that process. And we could see that in the normal mice, the expression of these genes is elevated during this daytime period. And that was lost in the mice that don't have a circadian clock in the liver, consistent with this idea that the liver clock basically functions to enable predicting that time and compensate for it by increasing the production of sugar in the liver. So now I'm going to shift and tell you about a recent story that came from my lab at Scripps. So I'll give you a little bit of background into how we got interested in the project, and then tell you some of the results. And finally, should still have some time for questions. I hope to wrap up in about 10 minutes. Okay, so coming back to this video of the sunflower, all of this research, discovery that clocks are present throughout the body, that they seem to enable the prediction of fluctuations in the external environment. All of that, and also that mice lacking a clock in the liver have altered blood glucose. All of these things together combine to suggest that the presence of circadian clocks in different types of cells sort of optimizes our physiology, optimizes the interaction of our physiology with our surrounding environment. And so that leads to a prediction that if your internal clock time is out of sync with the timing of rhythms in the external environment, that would be expected to be detrimental. And there have been a few groups that have done really elegant studies testing that idea in different organisms. And here on the left, I'm showing an example that was done in plants in a group led by Alex Webb. And what they did was they created genetic plants with genetic mutations that altered their internal circadian periods. So they, similar to the fruit flies that were identified by Ron Konopka and Seymour Benzer, they could identify plants that still had circadian rhythms, but with either a much shorter than normal period or a much longer than normal period. So here I'm showing in the paper that they published, they made mutants that had a 20-hour period or a 28-hour period. And then they could house these plants and expose them to light cycles that also had either a 20-hour period or a 28-hour period. So they would either have the lights on for 10 hours and off for 10 hours, or on for 14 hours and off for 14 hours. And when they grew these genetically altered plants in external environments that had a periodicity that it was either matching their internal timer period or different from it, you can see that the plants that are on this top left to bottom right diagonal have a genetic period that matches the external environment, and they are clearly healthier than the plants that have a genetic period, internal period, that is different from that of the external environment. So this was a really nice, elegant demonstration that the synchronicity between your internal and your external timers matters. And similar experiments have now been done in other organisms as well, but I think that's a really nice illustration of the point. So what about people? As we've talked about, our modern lifestyle involves several features that can be disruptive to our interaction with the light in our external environment. So you may have heard that light at night can be detrimental, and that is more and more common in modern life. Also, Jamie mentioned time shifts, and daylight saving time is coming up again. But in addition to that twice yearly shift, simply the definition of time zones can actually make a difference. And so around the world, how well the sort of official time is synchronized to solar time when the sun is directly overhead at noon is widely variable. And so here in this map, everything that's white is showing where the time zone definition is in sync with solar time. And in red is when the time is later than solar time, and in blue is when it's earlier than solar time. And a couple of different studies have actually demonstrated, with huge data sets, either in the US or a different study that, that had data from Russia and China, that living farther to the west in a time zone can actually be detrimental to your health and increase the risk of metabolic disease and cancer. And that is something that is not well understood, but is clearly present in the data. Oh, here's some of the data from the study that looked at cancer. And so for several different types of cancer, people who live further west in a time zone have an increased risk for several different types of cancer. But a more sort of dramatic desynchrony with the external environment comes from shift work, which is an essential feature of our modern economy. And that's something that we decided to study in my lab. So several different studies have shown that shift work can increase the risk of several different diseases. And as you might imagine, there's a huge amount of heterogeneity in this type of data. And so it can be very difficult to get a clear answer to this question. And there are lots of confounding factors, including diet and other lifestyle factors that can influence it. And so there is still debate over how big of an effect this is, but it's very clear for at least some types of cancer, including breast cancer, for which there's a lot of data, that long-term shift work does increase the risk. And so what we decided to do in my lab, we've been using a protocol to mimic a fairly severe shift work, rotating shift work schedule that we call chronic jet lag. In which, so again, as I've shown in a couple of different data slides, showing here how we set up the lighting schedule. And wherever you see a white bar, that's when the lights are on, and a black bar is when the lights are off. And I'm always showing 2 days in a row on each horizontal line, just because that makes it easier to see patterns in how the light schedule changes over time. So on the left here is a normal lighting schedule with 12 hours of light and 12 hours of dark every day. And then on the right is showing how we, the light exposures that we use for this chronic jet lag schedule. And it basically involves 3 times a week shifting the, turning the lights on 8 hours earlier. And that's also called a phase advance, when the lights are turned on 8 hours earlier. And so after a week, it comes back into sync with the original schedule. And this type of approach has been used by other groups as well. And several different studies have shown that in mouse models of breast cancer, liver cancer, and lung cancer, exposing them to chronic jet lag, either with this protocol or a very similar disruption of light exposure, increases their tumor formation. And we decided to use a mouse model of lung cancer that's similar to the one that was published by Felice Papagiannakopoulos in Tyler Jax's lab a few years ago. He showed that exposing this fairly severe lung cancer model to chronic jet lag increased their tumor burden. We just, in his model, which is rather severe, there are 2 different genes that are disrupted. So it's a pretty strong lung cancer phenotype. We decided to use a mouse model that only has one of those genetic mutations. So it's a more mild lung tumor, because we were hoping that we might have a greater window to see an effect of circadian disruption on that tumor model. And this project was led by Marie Pariolo, who was a postdoc in my lab until recently. She took a job at Vividian in San Diego. So Marie, as I mentioned, used a slightly less severe lung tumor model where she only has a mutation in the KRAS gene. And she housed those mice either in the control normal lighting conditions or chronic jet lag for 20 weeks. And then she measured how many lung tumors they got. And as we had kind of expected, she saw a much stronger effect than what the other group that studied the stronger lung tumor model had seen. She saw almost a 70% increase in the number of tumors that were formed in these mice that were exposed to chronic jet lag. And so after she discovered that there was this really strong effect, that gave us confidence that we might be able to understand what was happening at the molecular level, because she was seeing such a big effect. And so she took tumor biopsies from mice that were either housed in the normal letting conditions or chronic jet lag. And then she prepared RNA and sequenced all of the RNA from those tumors. And what she found here, this is something called a volcano plot, in which we're looking at how much of each gene is expressed in each sample. And then the y-axis is basically a statistical measure of significance. And so the higher the dot is, the more clear it is that there's a difference between the tumors taken from different conditions. And all these red dots are showing genes that were very highly significantly different in the two groups of tumors. And what we saw, to our surprise, is that she saw a clear signature for increased expression of genes that are turned on by heat in the tumors that she collected from mice exposed to chronic jet lag. And I mentioned before transcription factors that are really important in the circadian clock mechanism. And all of these genes, so HSP stands for heat shock protein, all of these genes are activated by a transcription factor called heat shock factor 1, or HSF1. So this really suggested that chronic jet lag somehow activates HSF1. And here I'm just showing some data from Marie confirming that that is true. So these are biopsies taken from lungs at different times of day from mice that were housed either in normal conditions or chronic jet lag. In the normal conditions, you can see that HSF1 has a nice sort of circadian rhythm. It's peaking in the dark nighttime hours. In the mice exposed to chronic jet lag, it's kind of on all the time. And I mentioned before that body temperature is one of the physiological phenomena that has a really strong circadian rhythm. And this timing of HSF1 expression is synchronized to the body temperature rhythm in mice. And it's consistent with the idea that this rhythm is affected by body temperature. And it suggested to us that maybe chronic jet lag is affecting body temperature rhythms. And that could be part of the explanation for why these genes are turned on. And I should mention that other groups, primarily coming out of Susan Lindquist's lab at MIT, have shown that HSF1 can enhance the growth of several different types of tumor. And so this could be the explanation for why we're seeing more tumors develop in these animals. So this suggested to us that maybe circadian disruption primarily alters body temperature. And this has not been studied in really large numbers of people yet, but there have been a few studies that have suggested that this is also the case in shift workers. And here at the top is the title of a paper that did show that core body temperature rhythms are altered in nurses who do shift work in a manner that would be consistent with our hypothesis. So finally, I just want to thank, first of all, the people in my lab. I mentioned that this work I really highlighted at the end was led by Marie Pariulot. And I'm also listing here all of the other wonderful people I've had the pleasure of working with at Scripps, either in my lab and also collaborators at Scripps and elsewhere. And I also really want to thank the generous funding we've received, both from the National Institutes of Health and also from several generous donors. And without further ado, I do want to save some time for questions from you.
All right, thanks a lot, Katja. That was terrific. The audience is really engaged. We've got more questions we can possibly answer. So we'll do what we can, but we do have a transcript of everything that you've typed in, and Katja will have the opportunity to respond offline once we get done today. So one of the questions that came up which I found interesting was, are the effects of shift work, cumulative, or is it reversible? And do you have data about that, or if you don't, maybe could you speculate about it?
So this is a really good question. It's not something I personally study, but other people do study this. And as I mentioned, there's a lot of heterogeneity, and there's even, due to that heterogeneity, you need huge data sets to get a really clear answer to this type of question. and I don't think there's a definitive answer to this question. The data that's out there suggests that it is cumulative. So people, the greatest increase in disease risk is seen in people who do rotating shift work for many, many years continuously. So it does seem that it's cumulative, but I can't say with certainty that, either way. But the
Right.
data is
Okay.
consistent.
That makes sense, but it's certainly, one wonders after hearing this. So you talked about the genes clock and period and a few other those, and I just wanted to confirm those are conserved in people and mice.
Yes.
And so these are general things that happen in all mammals. And so, but here's the question. So mice are nocturnal and we're diurnal, and so how do you switch the phase of the by 12 hours of the response to the daylight cycle?
Mm-hmm. So another fantastic question. And there is some data basically showing that the clock in your brain is set specifically by light. Light is the signal that sets the timing of your clock in the brain. But the timing of the clocks in other tissues, like the liver, for example, is not set by light. It's set by when you eat. And so you might have heard about research on time restricted feeding, time restricted eating. And that largely comes out of this understanding that circadian clocks in all your other organs are driven by when you eat. And so it's important to have kind of a consolidating eating time and fasting time every day to keep those clocks well synchronized.
You didn't mention this in your talk, but I noticed in the slide where you were showing the different tissues that they actually respond at different times, and so it's a bit more complicated than just daylight.
Yes, exactly.
Okay,
And
yeah.
this is something that people do not understand well, why different tissues timing is slightly different. Probably the mechanisms that set the time in different organs are different, and we don't understand that well.
So a lot of people ask this question in a very different way, so I'll sort of aggregate it. So what about the timing of medications and or treatments? Let's say you're getting treated for cancer or you were taking insulin. And so how do your circadian rhythms affect response to medications? And does anybody even think about that when they do a clinical trial?
Yeah, fantastic question. This is actually one of the things I was most excited to research when I started my lab at Scripps. We've published one study that looked at the timing of delivery of a specific diabetes drug, and it does seem to make a difference in some of the molecular responses to that drug. Way too early to say, clinical usage, but I think it's a really important question that needs to be addressed. Some of you might have read, there was actually an article in New York Times Magazine about this recently. It's a very important question. There are several groups that are interested in this and trying to move research in that direction. It is not very well studied, partly just because, clinical trials are huge and expensive already as it is. Adding another variable of time of day makes it additionally difficult, but it's a very important question.
Right. So what about cancer cells? So do they become deaf to all of these signals or hyper alert to the signals and
Yep, another great question. So for a while, people thought that cancer cells did not have circadian rhythms. That's not entirely true. They tend to have, many cancers don't have circadian rhythms, but it's certainly not a requirement to form a tumor to lose your circadian rhythms. I think that they tend to have less robust circadian rhythms simply because they tend to have a lot of mutations, and those may affect circadian rhythms in ways we don't understand well. How the genetic components of circadian clocks influence cancer growth is another area of intense research and something I'm very interested in. It turns out that it really depends on what tumor type you're talking about and what are the molecular drivers of that tumor and how those are affected by the clock. So you can't really make a blanket statement about how circadian rhythms affect cancer development.
Another area that a number of people were touching on was just latitude. And what's the effect of being, I mean, if you're at a high latitude, then the days naturally go from short to long at a pretty amazing swing. Whereas if you're at the equator, it's much less. And so clearly our bodies adapt to the seasonal variation in daylight, but there's big differences. And so how does that work?
Yeah, all these things are just great questions. I don't have all the answers. This is not my area of expertise, but there are other people who work on this. And so first of all, latitude does affect things. I mean, there's something called seasonal affective disorder that affects people more and more the further from the equator they live, don't get enough sunlight during the day. And that affects, hormone production and all these different things. It's not my area of expertise, but that's definitely something that other people work on. And in other organisms, there is some research into how latitude affects biology. So you can imagine, in people, it's probably, we haven't evolved enough, with like clear populations in different latitudes to have a genetic study, but some other species have. And so, for example, in fruit flies, there are many different species of flies that live at different latitudes. And there are some genes that are made differently, spliced differently in species, subspecies of flies that live at different latitudes. So there is some effect there on clock genes as well.
Maybe you could say there's a lot of interest in the intersection of circadian rhythms with diseases. So are there specific diseases? Maybe you mentioned one, I think, in the last answer, but are there specific diseases? And what about insomnia? Is that intersecting with these pathways, or is that something else?
So yes, I think it intersects, but again, this is not my area of expertise, but so for example, light exposure has a really heavy influence on the production of a hormone called melatonin that's really important for sleep. And so the timing and intensity of that light exposure has a big impact on how much melatonin is produced. So it's kind of a combination of, you want to get more exposure to natural intense sunlight during the day, less exposure to light, and in particular blue light at night, to optimize the melatonin production. And that's work of other people, but yeah, very important for sleep. And lots of other things I'm sure contribute as well, but not my area of expertise.
So we're coming up on the hour, so I'm mindful of the time, and we have to draw this to a close. And I'll just ask one more question, which I found kind of amusing, and it's, can you influence legislation about daylight savings time?
So actually, well, go ahead. So there's a society called the Society for Research in Biological Rhythms, of which I am a member and have been for a long time. And we did put out a white paper, and we do have a committee that is trying to influence that legislation. And the evidence strongly supports that we should stop shifting our clocks. Importantly, we should have permanent standard time, not permanent daylight saving time. Even though it's really lovely to have, more light after work, it's actually the light exposure in the morning that's more important for properly synchronizing to the environment and getting enough.
I mean, the profound impacts of that simple change would be remarkable. So thank you for that work. Okay, we need to draw it to a close. There are tons of questions.
I can see there's a long list. I'll try to look them over.
They're still scrolling it. So this is great. We'll try to get back to that. And I just want to thank you for a terrific lecture, and everybody's clearly pretty engaged, and it's a subject we can all relate to. So thanks, Katya. And before I sign off, let me just remind you that the next front row is November 16th. That's going to be our colleague Silke Paust, and she's going to be talking about how the immune system attacks tumors and supercharging it to do so. So we hope to see you again at the front row. Thanks all for tuning in, and have a great day. Bye-bye.